Hey everybody, welcome to the last module in our electrophysiology portion of Module 7. So Module 7.2.5, and here we're going to talk about synapse. So in the nervous system, messages move from one location to another in the form of action potentials, like we talked about in the last module, or nerve impulses along the axon. And to be effective, a message must not only be propagated along the axon, but also be transferred in some way to another cell. This transfer takes place at synapses. A synapse may be electrical, with direct physical contact between the cells, or chemical, involving a neurotransmitter. At electrical synapses, the presynaptic and postsynaptic membranes are locked together at gap junctions. The lipid portion of the protein membranes, separate only by about 2 nanometers, are held in position by binding between integral membrane proteins called connexons. These protein forms pores that kind of permit ions to pass between the cells. But because the two cells are linked in this way, changes in the membrane potential of one cell produce local currents that affect the other cell as if the two shared a common membrane. And as a result, the electrical synapse propagates the action potential quickly and efficiently from one cell to the next. Electrical synapses are extremely rare in both the CNS and the PNS, and they occur in some areas of the brain, including the vestibular nuclei, the eye, and at least one pair of the PNS ganglia, the ciliary ganglia, to be exact. The situation at a chemical synapse is far more variable than that of an electrical synapse because the cells are not directly coupled. For example, the action potential that reaches an electrical synapse is always propagated to the next cell, but at a chemical synapse, an arriving action potential may or may not release enough neurotransmitter to bring the postsynaptic neuron to threshold. In addition, the other factors may intervene and make the postsynaptic cell more or less sensitive to arriving stimuli. In essence, the postsynaptic cell at a chemical synapse is not a slave to the presynaptic neuron, and its activity can be adjusted or tuned by a variety of factors. So, chemical synapses are by and far the most abundant type of synapses in the body. Most synapses between neurons and other communications between neurons and the other types of cells involve chemical synapses. Normal, Normally, communication across chemical synapse takes place in only one direction, from the presynaptic membrane to the postsynaptic membrane. So, acetylcholine, or ACH, is the neurotransmitter that has received most of our attention so far in this unit, but there are other important chemical transmitters. Based on the effects of the postsynaptic membranes, neurotransmitters are often classified as excitatory and inhibitory. Excitatory neurotransmitters cause depolarization and promote the generation of action potentials, and inhibitory neurotransmitters cause hyperpolarization and suppress the generation of action potentials. This classification is useful, but not only always precise. For example, acetylcholine typically produces a depolarization in the postsynaptic membrane, but ACH releases a neuromuscular junction in the heart has an inhibitory effect, produces a transient hyperpolarization of the postsynaptic membrane. This situation highlights an important aspect of neurotransmitter function, which is the effect of a neurotransmitter on the postsynaptic membrane depends on the properties of the receptor, not on the nature of the neurotransmitter itself. So synapses that release ACH are known as cholinergic synapses. The neuromuscular junction is an example of a cholinergic synapse. ACH is the most widespread and the best studied transmitter. It's released at 1, all neuromuscular junctions that involve skeletal muscle fibers; 2, at many synapses in the CNS; 3, at all neuron-to-neuron synapses in the PNS; and 4, at all neuromuscular and neuroglandular junctions in the parasympathetic divisions of the ANS, which we'll get to in a few modules. At a cholinergic synapse between two neurons, the presynaptic and postsynaptic membranes are separated by a synaptic cleft that averages about 20 nanometers in width. Most of the ACH in the axon terminal is packed in synaptic vesicles, each containing several thousand molecules of the neurotransmitter. And a single axon terminal may contain millions of vesicles. So, getting to the events at a cholinergic synapse. In step 1, an action potential arrives and depolarization of the axon terminal. The normal stimulus for a neurotransmitter releases its depolarization of the axon terminal by the arrival of that action potential. Step 2, or the extracellular calcium ions enter the axon terminal, triggering the excitosis of ACH. The depolarization of the axon terminal briefly opens its voltage-gated calcium channels, allowing calcium to rush in. Their arrival triggers exocytosis of ACH into the synaptic cleft. The ACH is released into packets of roughly 3,000 molecules or so, and the average number of ACH molecules in a single vesicle or so. ACH release stops very soon because active transport mechanisms rapidly remove the calcium ions from the terminal cytosol. These ions are either pumped out of the cell or transferred into the mitochondria. In step 3, ACH binds to receptors and depolarizes the postsynaptic membrane. So ACH then diffuses across the synaptic cleft towards the receptors on the postsynaptic membrane. These ACH receptors are chemically gated ion channels, and the primary response is an increase in permeability to sodium, which produces a depolarization in the postsynaptic membrane that lasts about 20 milliseconds or so. These cation channels also let potassium ions out of the cell, but because sodium ions are driven by a much stronger electrochemical gradient, the net effect is a slight depolarization of the postsynaptic membrane. This depolarization is a graded potential. The greater the amount of ACH released at the presynaptic membrane, the greater the number of open cation channels in the postsynaptic membrane, and so the larger the depolarization. If the depolarization begins in an adjacent area of the excitable membrane, such as the initial segment of the axon, to threshold, an exponential appears in the postsynaptic neuron. In step 4, ACH is removed by acetylcholinesterase. The neurotransmitter effects on the postsynaptic membrane are temporary because the synaptic cleft in the postsynaptic membrane contains the enzyme acetylcholinesterase. Roughly half the ACH released in the presynaptic membrane is broken down before it reaches the receptors on the postsynaptic membrane. ACH molecules that bind receptor sites are generally broken down within 20 milliseconds of their arrival. So acetylcholinesterase breaks down molecules of ACH by hydrolysis into acetate and choline. The choline is actively absorbed by the axon terminal and is used to synthesize more ACH using acetate provided by coenzyme A. Acetate diffusing away from the synapse can be absorbed and metabolized by the postsynaptic cell or by other cells and tissues. And then this slide just kind of overviews of what we just talked about. So the nervous system relies on a complex form of chemical communication. Each neuron is continuously exposed to a variety of neurotransmitters. Some usually have excitatory effects and others have inhibitory effects. Yet, in all cases, the effects depend on the nature of the receptor rather than the structure of the neurotransmitter itself. And many drugs affect the nervous system by stimulating receptors that otherwise respond only to neurotransmitters. And these drugs can have complex effects on perception, motor control, and emotional states. Major categories of neurotransmitters include biogenic amines, amino acids, neuropeptides, dissolved acids, and a variety of other compounds. Here we're going to cover some of the common neurotransmitters that we'll see, and I've included a chart of some of the major ones that you can see here, but I'm just going to highlight a few. We've already talked about ACH extensively here. We know that ACH is excitatory, has a direct effect, and it affects the skeletal muscular junctions, many CNS synapses, and all neurons of the PNS, as well as the parasympathetic ANS. But to highlight some of the others that we haven't talked about as much, norepinephrine, or NE, is a neurotransmitter that is widely distributed in the brain and other portions of the NS. Norepinephrine is also called noradrenaline, and synapses that release NE are known as androgenic synapses. Norepinephrine typically has an excitatory, depolarizing effect on the postsynaptic membrane, but the mechanism is quite distinct of that of ACH. Some of the others that we haven't had the chance to cover as much. So dopamine is a CNS neurotransmitter released in many areas of the brain. It may have either inhibitory or excitatory effects. Inhibitory effects play an important role in our precise control of movement. For example, dopamine releases in one portion of the brain prevents the overstimulation of neurons that control skeletal muscle tone. If the neurons that produce dopamine are damaged or destroyed, the result can be characteristic rigidity and stiffness that's seen in Parkinson's disease. Which is a condition that we described later on. In other sites, dopamine release has an excitatory effect. So cocaine inhibits the release of, or inhibits the removal of dopamine from synapses in specific areas of the brain. and the resulting rise in dopamine concentrations at these synapses is responsible for what we consider the high that is experienced by cocaine users. Serotonin is another important CNS neurotransmitter. Inadequate serotonin production can have widespread effect on a person's attention and emotional states and may be responsible for many cases of severe chronic depression. So fluoxetine, or better known as generic name Prozac, paroxetine or Paxil, and sertraline or Zoloft, and related antidepressant drugs inhibit the reabsorption of serotonin by axon terminals, hence their classification as selective serotonin reuptake inhibitors, or SSRIs. This inhibition leads to an increased serotonin concentration at synapse, and over time, the increase may relieve the symptoms of depression. Interactions among serotonin, norepinephrine, and other neurotransmitters are thought to be involved in the regulation of sleep and wake cycles. The last one I kind of want to hit here is the gamma aminobutyric acid, or GABA, and generally has an inhibitory effect. Roughly 20% of the synapses in the brain release GABA, but its functions remain incompletely understood. In the CNS, GABA release appears to reduce anxiety, and some anti-anxiety drugs work on enhancing this effect. So the functions of many neurotransmitters are not well understood. They kind of just happen. We don't really know why. In a clear demonstration of the principle, the more you look, the more you see. Over 100 neurotransmitters have been identified, including certain amino acids, peptides, polypeptides, prostaglandins, and ATP. In addition, two gases, nitric oxide and carbon monoxide, are known to be important neurotransmitters. Nitric oxide is generated by axon terminals that innervate smooth muscles in the walls of the blood vessels in several regions of the brain. And carbon monoxide, best known as a component of the automobile exhaust, is also generated by specialized axon terminals in the brain, where it functions as a neurotransmitter in the PNS and its synapses. And these can have either a direct effect on the membrane potential, either it closes or opens the ion channel and provides a rapid response, or it can have an indirect effect on the membrane potential, meaning that it binds the receptor and activates the G protein. And active G proteins activates will second messengers, whether they be cyclic AMP or CGMP or diacylglycerides or calcium channels. And the second messenger ion channels or active enzymes, it provides a slower but longer lasting effect. For example, norepinephrine in androgenic synapse. And examples of indirect action, the neurotransmitter will bind to the receptor activates the G protein, the G protein activates adenyl cyclase, and adenylate cyclase converts ATP into cyclic AMP, and CCAMP opens the ion channels. Postsynaptic potentials are graded potentials that develop in the postsynaptic membrane in response to a neurotransmitter. Two major types of postsynaptic potentials developed at neuron-to-neuron synapses, or excitatory postsynaptic potentials and inhibitory postsynaptic potentials. An excitatory postsynaptic potential, or an EPSP, is a graded depolarization caused by the arrival of the neurotransmitter at the postsynaptic membrane. An EPSP results from the opening of chemically gated membrane channels that lead to a depolarization of the plasma membrane. For example, the graded depolarization produced by the binding of ACH is an EPSP. Because it's a graded potential, an EPSP affects only the area immediately surrounding that synapse. And we've already noted that not all neurotransmitters have an excitatory or depolarizing effect. An inhibitory postsynaptic potential, or an IPSP, is a graded hyperpolarization of the postsynaptic membrane. For example, an IPSP may result from the opening of a chemically gated potassium channel. While the hyperpolarization continues, the neuron is said to be inhibited because a larger than usual depolarization stimulus is needed to bring that membrane potential to threshold. A stimulus that shifts the membrane potential by 10 millivolts would normally produce an active potential, but if the membrane potential were reset at minus 85 millivolts by an IPSP, the same stimulus would depolarize only to about minus 75, which is below that threshold needed to propagate an active potential. An individual EPSP has a small effect on the membrane potential itself, typically producing a depolarization of about 0.5 millivolts at the postsynaptic membrane. Before an active potential will arise in the initial segment, local currents must depolarize to that region by at least 10 millivolts. Therefore, a single EPSP will not result in an action potential, even if the synapse is on the axon hillock. But an individual EPSP combined through the process of summation, which integrates the effects of all the graded potentials that affect one portion of the plasma membrane. The graded potential may be EPSPs, IPSPs, or both. And we'll consider EPSPs in our discussion that we're talking about here. Two forms of summation exist. We have temporal summation and spatial summation. Temporal summation is the addition of the stimuli occurring in the rapid succession at a single synapse that is actively repeated. This form of summation can be likened to using a bucket to fill a bathtub. You can't fill the tub with a single bucket of water, but you will eventually fill it if you keep repeating the process. In the case of temporal summation, the water in the bucket corresponds to the sodium ions that enter the cytosol during the EPSP. A typical EPSP lasts about 20 milliseconds, but under maximum stimulation, an action potential can reach the axon terminal each millisecond. Every time an action potential arrives, a group of vesicles discharges ACH into the synaptic cleft. And every time more ACH molecules arrive at the postsynaptic membrane, more chemically gated channels open, and the degree of depolarization increases. In this way, a series of small steps can eventually bring the initial segment to threshold. Spatial summation occurs when a simultaneous stimuli applied at different locations have a cumulative effect on the membrane potential itself. In other words, spatial summation involves multiple synapses that are actively simultaneous. In terms of our bucket analogy that we just talked about, you can fill a bathtub immediately if 50 friends kind of empty their buckets all at the same time. In spatial summation, more than one synapse is activated at a time, and each pours sodium ions across the postsynaptic membrane, producing a graded potential with localizing effects. At each active synapse, the sodium ions that produce the EPSP spread out along the inner surface of the membrane itself and mingle with those entering at the other synapses. As a result, the effects of the initial segment are cumulative, and the degree of depolarization depends on how many synapses are active at any moment and their distance from the initial segment. As in temporal summation, an action potential results when the membrane potential at the initial segment reaches that threshold. So here we'll talk about facilitated, which can be considered depolarization, and the postsynaptic potentiation. So it's convenient to discuss each synapse as if it were releasing only one chemical, but axon terminals may release a mixture of active compounds either through diffusion across the membrane or by exocytosis, along with neurotransmitter molecules. And these compounds might have a variety of functions, those that alter the rate of neurotransmitter release, or by the postsynaptic neuron, or change the postsynaptic cell's response to neurotransmitters. And this is what we see at the bottom of the line here, are the neuromodulators. And these substances are typically neuropeptides, small peptide chains, and synthesides, and released by the axon terminal. And most neuromodulators act by binding receptors in the presynaptic or postsynaptic membranes and activating cytoplasmic enzymes. Neuromodulators called opioids have effects similar to those of the drugs opium and morphine because they bind to the same group of postsynaptic receptors. There are four classes of opioids in the CNS. We have endorphins, enkephalins, endomorphins, and dimorphins. And the primary function of opioids is to relieve pain. And they inhibit the release of neurotransmitter substance P at the relay pain sensations. Dynorphins have a far more powerful pain relieving effects than morphine and other opioids. Endorphins are so named because they act like endogenous morphine. And in general, neuromodulators have long-term effects that are relatively slow to appear, and two, trigger responses that involve a number of steps and intermediary compounds. Three, they may affect the presynaptic membrane and the postsynaptic membrane, or both. And finally, four, can be released alone or with a neurotransmitter. And in practice, it can be very difficult to distinguish neurotransmitters from neuromodulators, either biochemically or from a functional basis. And a neuropeptide may function at one side as a neuromodulator, at another side as a neurotransmitter. So it's kind of difficult to decide here. Inhibitory or excitatory responses may occur not only at synapses involving the cell bodies and dendrites, but also at synapses found along the axon or its collaterals. At an axonomic or axon to axon synapse, a synapse occurs between the axons of two neurons. An axo-ionic synapse at the axon terminal can either decrease, inhibit, or increase, facilitate the rate of the neurotransmitter release at the presynaptic membrane. In the presynaptic facilitation, activity at an axo-ionic synapse increases the amount of neurotransmitter released when the action potential arrives at the axon terminal. This increase enhances and prolongs the neurotransmitter's effect on the postsynaptic membrane. The neurotransmitter serotonin is involved in presynaptic facilitation. In the presence of serotonin, released at an axo-ionic synapse, voltage-gated calcium channels open and remain open longer. In one form of presynaptic inhibition, the release of GABA inhibits the opening of voltage-gated calcium channels in the axon terminal, and this inhibition reduces the amount of neurotransmitter released when the action potential arrives there, and thus reduces the effects of the synaptic activity on the postsynaptic membrane. There are some factors that disrupt this neurofunction. pH has an effect, the ion concentration, temperature has an effect, nutrients have an effect, and oxygen also has an effect.